There we go. Okay. Thanks everyone for joining us today. Again, I just want to quickly mention that we're using a webinar. So if you'd like to ask a question, please use the raise hand button and then I will unmute you so that you can just directly ask the question to our speaker. So it's my pleasure to introduce Glamina. She's a professor in the Department of Biology here at Brandeis University. Her research focuses on identifying the key physical properties of individual cilia that control the collective phenotypes of ciliary array and the structure of the flow it generates. So today she will share with us some of the related work about cilia-driven flows linking microscopic properties with emergent macroscopic dynamics.
Hi everyone. Thanks for hosting me. And then, yes, there we go. As you mentioned, feel free to interrupt at any point if you have any questions about anything. And then today I'll share with you some work I did in my postdoc, trying to understand how do we connect the activity of a relatively small length scale cellular organelle with the larger macroscopic properties of the flow we see here. And then I'll also share with you some of the directions that I'm currently working in my lab towards understanding this problem in a more broad context in different organisms and biological systems. So as I mentioned, the problem is how do we build a macroscopic flow in a biological system if we have individual units? And before I go into the nitty gritty of this, I just want to show you what do I mean by biological flows. In this case, we see examples of flows that are being generated by different organisms across the tree of life. And all of these flows are generated by arrays of motile cilia. And what I want to draw to your attention to in this slide is the diversity of architectures we can already see in these different organisms. But then of course, it's not so surprising if you think about why they need to generate these flows. So if, in this case, if you're a single cell that's swimming in a pond, you probably need flow to swim and trap particles so you can feed. So that's going to be a very different flow topology than where you're going to want to generate if you're an airway. In this case, the airway needs to generate a flow that clears mucus from the lungs to the mouth. So then you would want to avoid vortices and things like that, like in this case. But what's really interesting is that in all these biological systems, what's actually driving these flows that appear very different is the same cellular organelle. So here I'm just showing you the activity of a single cilium. Like this is a single cilium in all its glory. And I want you to appreciate as well the length scale. So here we have a cell, and then cilia are the cellular appendages that are like long filaments. And they generate flow by executing this periodic beating cycle. So then as you can see, there's an active stroke and then a recovery stroke. And then the asymmetry of this stroke is actually what's driving flow at this lower Reynolds number, because again, the length scale in this case is each cilium is 10 microns. And then I want to emphasize again the difference in length scales that we have here. So here we have a cilium that is 10 microns in length. And the flows I showed you were centimeter scales flows. And then the question is really, how do we understand the flows that this is generating if there's such a large length scale in disconnect? So then how cells achieve this is actually they pattern hundreds of cilia, thousands of cilia, to generate the particular flow architecture that I showed you. So throughout different biological systems, we can see the differences in how systems arrange their cilia. In this case, it's a single algae and it's swimming around, so it only needs 2 cilia, but then you can get more and more complex. So you get this free living unicellular cells that have thousands of cilia within one cell. But then again, it's only one cell. And then you can also arrange your cilia in a more complex way. So you have a tissue, like the case of the airway in this case, or this planaria flatworm. This tissue is going to have cells that are ciliated. So they have cilia, but then you're going to have cells that don't have cilia. So what my group is trying to do is try to understand how by patterning the cilia in an array, we can give rise to these different flow topologies that we see. And then what properties really of the individual filaments are actually going to control the dynamics of the flows we see generated in all these cases. And I want to emphasize again that this is a problem where we have a variety of length scales that might be important because we have a single cilium that the activity of a single cilium is coordinated by arrays of molecular motors that are organized at the nanometer scale. And then if we increase our length scale, we can see that there's different types of organization that are gonna be important. And all of this actually feed into the larger structure of the flow at the macro scale. We have different asymmetries, they're all these different length scales. We can start looking at an individual cilium. I showed you it has an asymmetry in its beat waveform, but it also has a spatial asymmetry. So it has a well-defined plane of beating. So this cilia are beating in one plane, and the plane of beating of each cilium is gonna be determined by the relative orientation of these two different structures in the membrane. So this structure anchors the cilium to the cell membrane, and then the relative orientation of this basal fluid and basal body is gonna define the plane of beating of every cilium. So this already shows that we have an asymmetry at the level of one single organelle. But we can think that we also have organization at the scale of a cell. So in this case, if we have one cell and each cell has tens or hundreds of cilia on its surface, then the relative orientation between the neighboring cilia is gonna become important for the direction of flow. And now we increase complexity again, and we think about a tissue that has different cells, and then cells need also to be oriented relative to a particular tissue axis. We get another tissue scale asymmetry, where we can see in this case is some fluorescent staining of a protein that gets segregated to a particular side of the cell, and that signal tells the cilia to orient relative to that direction. So what I really try to understand is how properties that or asymmetries that occur at this scale feed into the different length scales. And then how independently each one of these types of spatial organization and patterning gives rise to the particular properties of the flows that we have. And so it's a question about connecting the length scales and which one is important in different contexts. And today, most of the talk is gonna be focused on this mesoscale and try to understand how the architecture of how I pattern the cilia at the tissue scale is gonna give rise to a flow with a particular topology and that is tailored to execute a very specific function. In this case today, I'm gonna be talking about the airway. So in the airway, we have this tissue that is composed of multiciliated cells. So every single cell, like in this cartoon, has hundreds of cilia on its surface. And then in this, since we have certain biological constraints, the airway needs to execute mucus clearance. So we have a mucus layer that is trapping all the pathogens, and then that needs to be carried away from the airway. So we need other cells that secrete the mucus. We can't have just a surface that is uniformly covered by cilia. And then we have, in the end, we end up with a patchwork of multiciliated cells and other cell types. So not all of the cells are gonna have activity. And that's gonna become very important when we try to understand how flow is generated. And here I want us to stop for 1 second to think about how important the function of this is. Here what I'm just showing you is a movie of a piece of mouse airway, but this is what's happening in our airway right now. If we just stop and take a deep breath, we have each one of these units is a multiciliated cell that has hundreds of cilia and that are constantly beating to clear mucus. And then you can get a full appreciation of the problem here. You have this tissue that is hundreds of microns and each cell has cilia that need to coordinate and to make mucus clear, like let's say in this case from here to here, but across the whole tissue. So the question becomes, how is this achieved? And when we think about what would be important to generate a flow that goes from the lungs to the mouth, we can stop and think about what are the biological constraints that we have, and then which parameters would be important for designing an airway that is able to execute this function. We know that the driving activity of what's gonna drive the flow is gonna be the activity of the cilia that are organized in multiciliated cells. So we know that like how they are patterned is gonna be important. And what else do we know? We know that we're not gonna be able to have a carpet, a continuous tissue with cilia, because we have other cell types. So we know that the coverage fraction of this tissue by multiciliated cells is going to be less than 1, just because we need other cell types in this case. And then, so we have to be, well, then they suggest that how we arrange them is going to be very important, right? So if we have them all clustered in here, it might be that this activity is not sufficient to generate a flow that will move things from here to here. So then perhaps we need to arrange them in some optimal way for them to be able to connect flow from here to here. So then I'm gonna be showing some measurements of the relative distribution of these multiciliated cells, and then I'm gonna define this wavelength as the average distance between cell clusters. And then that's gonna be an important parameter of how this airway generates this connected flow. Then we have a wavelength, and then I also told you that individual cilia have a plane of beating, and then also we can think about each cell has an average direction in which it's propelling fluid. So then this is gonna be described by an orientation. So intuitively, we can think that, okay, if I need to generate a globally connected flow from here to here, then the cilia should be oriented parallel relative to each other in order to generate this flow. And then they should be pushing things in the same direction. So then this is gonna be described by an orientation. And I'll show you some measurements from a biological system that we know is able to clear mucus of these different parameters. And then we can try and see how a biological system achieves a globally directed and coordinated flow given the constraints it has. Here, what I'm just showing you is microscopy data of the airway. So as I mentioned before, this is all data from a mouse. What I did was I cut out the trachea, the entire trachea of the mouse, and then I cut it open lengthwise. And then you put it in a cover slip, and then you can image every single cell that has cilia at the scale of the entire airway. So we have that this is the entire trachea. It just looks like a sea of green. Here, the multiciliated cells are tagged with a green marker. But then when you zoom in, you can really see that this is more like a patchwork pattern. So you have this green that tags the multiciliated cells, but then you also have this space where you have other cells. And again, the activity is purely driven by this green cell. So wherever we have blood, we don't have green cells, then there's no activity. And so we can actually measure from data like this, the parameters I showed you. So first, we measure the coverage fraction. And you can see that there's certain differences depending on which region of the airway you are. But if you take an average of it, we see that the coverage fraction is around 0.4. So what this means is that less than half of the surface of the tissue is actually covered by multiciliated cells. So this was kind of surprising at the beginning because you would think that if you need a flow that is globally connected and directed in this one direction, you would need more of a continuous carpet, but apparently you can do it without less than half of the total surface being covered by ciliated cells. And then from data like this one on the right, you can measure something like the average separation within these clusters. So here what I'm just showing you is an autocorrelation function of the pattern. And then from this, you can get the average separation between green clusters in this image. And when you take the average over several tissue sections, you can see that it's 16 microns. So this corresponds to roughly 3 multiciliated cells. So even if the cells are themselves don't have a high coverage fraction, they are distributed in a way that they're actually in close proximity to each other. You never see a gap that's bigger than 3 to 4 cells on average. So this already tells us how would you build, how an airway builds this directed flow. And then finally, I want to show you some measurements of the orientation. So the orientation, as I said, at the individual cilia level is defined by the relative positioning of this basal body and basal foot. So if we zoom out, this is an entire section of tissue, and then in blue, we can see the cell membranes. So each one of these little hexagons is a multiciliated cells. And then here the black regions are just other cells that don't have cilia. And then here you can see that each one is a cell, but then when you really zoom in, you can start to see this dotted pattern. So each one of these dots corresponds to an individual cilium. So if you do measurements of how many cilia you have per cells, you have anywhere from like 80 to 300 or 400 or so. And then so the cell is completely covered by cilia, but then when you zoom in even further, you can start to see these dots, right? So we're able to actually resolve the green from the red. And then what we do now is we just connect the dots. So every single one of these arrows represent the orientation of an individual cilium. So we can do this measurement for all the cilia within every cell. Then we get a polarity field. So for every cell, we get the orientation of individual cilia, and then that way we can describe in which plane each cilium is feeding. And then already from here, you can see that this was counterintuitive a little because you see that there's defects in the orientation of cilia. So from the literature, people thought that like, if you have an array of cilia, they all better be having the same plane of beating and oriented in the same direction relative to each other if you want to generate a coordinated flow. But here we see that that is not the case. And then in fact, there's a level of heterogeneity. And then we see also these defects that might have to do with the actual constraints on the cell shape and the way you can pack the cilia that itself doesn't allow for all the cilia to be oriented in the same direction and be perfectly parallel. But then we were interested in the tissue scale. And in this case, so what I did was, since we want to look at many cells across the entire tissue, we can just calculate the average orientation of the cilia within a cell, and then assign that to every cell in the tissue, and we end up with something like this. So this is the polarity field that describes the orientation of every cell in this tissue. And then you can see already just from this one example that the heterogeneity is present as well at the tissue scale. And then there is some level of relative disorganization, at least across the entire tissue section. And we can measure this just by defining an order parameter, much like magnetization. And then we can see that at the cell scale, we see cells that have a high level of disorganization all the way from like very low values to almost perfect orientation. So there's heterogeneity at that level, but then that all gets smoothed out at the tissue scale. However, we do see a tissue where the cilia are not all oriented perfectly parallel to each other, which was kind of surprising because intuitively, like I mentioned before, to build a directed flow, you would need all like the active units that generate the flow be pushing in the same direction. And then also we know that in other biological systems, this is also possible. So in cases where you have pattern of hairs and things like that, they're all like way more oriented relative to each other. So this suggested to us that maybe it's not that this tissue can't achieve this because of the biological constraints, but rather it's building in like some heterogeneity on purpose, let's say. So I want to conclude this section by just summarizing what I just showed you. I showed you the coverage fraction of multiciliated cells in the airway of a mouse. It's around 0.4, so less than half of the total surface is covered by multiciliated cells. But then the gaps between multiciliated cells are not so large. In fact, they're around 16 microns, so 3 cells are at most. And then perhaps the most surprising measurement was that if we look at the orientation of the cilia within multiciliated cells, we do measure heterogeneity in that. So then the question becomes like, how from this heterogeneity we can achieve an organized and connected flow. And then here, I think it's a good point to pause and see if anyone has any questions or clarifications.
I'm curious, so you mentioned that maybe there's some constraints on the surface of the cells that require a certain number of defects. And this vector field. Is the shape of those cells very uniform? I mean, do you get kind of a constant curvature, or?
So in these cartoons, they're perfect hexagons. I haven't really explored this, but just qualitatively, they're never this case. And there's like, if you just look at, you know, the data, even in this case, you can see that the shapes are very heterogeneous. So then I think that's a cool question. That from this data, you can also measure, you know, what type of polygons you get, and then how does that relate to the defects you see in the cilia packing. So people have shown that the number of cilia that each cell has is proportional to the surface area of the cell. There's nothing by itself like saying, oh, the shape should constrain that. But then my guess is that the shape is actually restricting how you pack your cilia. So then you would expect certain defects from different cell shapes. So that's something interesting that you can also get from here, for sure. Then, so I'll just show you then, you can think, okay.
Sorry, we do have one more question. Yeah. Okay, so Peter, go ahead.
Hey, how's it going? So, okay, quick question. So you're mentioning that, I guess in the lungs, the purpose of these flows is to clear mucus. And then, you know, there's surprises based on, I guess, the disorder and it not being, people thought you needed ordered ciliary networks to generate flow. But like, where does the mucus go? Like you would want coherent flows if it's all like, you know, going to go in one direction. But yeah, I guess, how do you clear the mucus?
It all goes in one direction because it goes from the lungs to the mouth. So that's where it goes.
I see. So it just all comes out the mouth.
Yeah, exactly. It comes up the mouth and then to the, like from the lower to the upper respiratory tract. So you want something that's highly directed in the specific case of the trachea. And then here's what this looks like, actually in this case of the mouth. So also, yeah, you can see that here it was connected to the lungs and then here's the mouth. And then you can see that that's where all the mucus is going because the whole function of this is to provide some immunity to the airway. So now it's like this thing is very in vogue, but like the idea is like you have mucus, it traps all the pathogens and particles, and then it has to be cleared out. So that's why it goes kind of from the lower to the upper respiratory tract. And
awesome.
Then,
thank you so much.
Yeah, sure. And then, yeah, so you don't want to have like these regions where things accumulate and like, you know, then particles and pathogens would like invade the cells, et cetera, et cetera. So you want to have a flow that is highly directed. And then here what we're seeing is the flow across the entire trachea. So then again, I want to point out the scale bar. This is the entire trachea. And then globally, we have a very directed flow that goes in one direction. And then of course we can measure this and do PIV to get the whole velocity field. And then we can confirm with that actually the correlation length of this flow is like the entire trachea. So from that, the question becomes like, okay, then if I have this globally directed flow that is highly correlated a lot across this entire length scale of the organ, then how can I build that if I have a disordered tissue? So to investigate this question, what we did is we turned to modeling. And then there's several ways you can model cilia from the like most complexity to the least complexity. Here we're interested in this macroscopic scale. So we're not gonna worry about like modeling explicitly the beat of every single cilium or the activity. Rather we say, okay, if we have a piece of tissue and then we have multiciliated cells, we're just gonna introduce the activity of every multiciliated cell as a slip velocity at the boundary between the tissue and the fluid. And then we're gonna see if we have different arrangements of multiciliated cells, what can we expect, right? So if we have here the clusters of cells and we vary the separation between them. So this was described by the wavelength previously, but then here we want a dimensionless parameter. So we have the wavelength divided by the height of the fluid film. And then that's gonna describe how separated the clusters are. And we can ask, okay, as a function of that separation, what type of flow architecture we expect to get. And then also what type of, and then as I mentioned as well, the orientation was the most puzzling part. So then here would be really interesting to see if we have a perfectly oriented network, then how would that be? What type of flow architecture would that generate, and how does that compare to the actual level of disorder we see in the tissue? And then another interesting thing here was that to describe flow, from what I showed you, what's clearly more important is not really just how much flux goes through the layer, but rather also the topology and structure of the flow. So we wanted a way to distinguish between a flow that is unidirectional versus something where you have these regions of recirculation and things start accumulating. So we came up with this clearance time, which is basically a mean exit time. If you drop a bunch of particles, how long do they take to clear out from this tissue piece? But then I'm actually interested if anyone has any opinions or suggestions of how to define quantitatively flow topologies, because this is something that's very specific to the airway, but it's harder to generalize to other things. But here I'll just show you in terms of clearance time, what happens as we vary the architectures of the tissues. And here I'm just showing you what happens with this patchiness parameter. So the separation between the clusters. And here, these green arrows represent the multiciliated cells. So here we have the same coverage fraction in all of these cases. So we have the same number of multiciliated cells. All that we're varying is how we distribute them in the tissue. And then you can already see that this makes a huge difference. If we have a large cluster like here, or even like if we split it into several clusters but that are relatively large, instead of spacing them together and having smaller clusters, then we see these regions of recirculation emerge. But you can see that as you make smaller clusters of cells that are closer together, then you're able to get rid of these regions of recirculation. So the airway lives somewhere around here, where you have, even though your coverage fraction is relatively low, the separation between the multiciliated cells is not so big. And then here we can do a full parameter sweep, and then we can see that the total flux only depends on the coverage fraction by the cilia. So it's gonna be the same in all these cases. However, the clearance time gets reduced when we reduce the average separation between clusters of multiciliated cells. So then this again shows the importance of finding the right parameters to characterize the flow. And here I'm showing you what happens as a function of orientation, which was perhaps the most intriguing parameter. So in all of these cases, we have regions of recirculations because we're in that region of phase space. And then here we have perfect orientation. And then we start to increase the disorder. And then qualitatively, you can already see what would be the physical mechanism by which disorder is helping you. Do you have a question, Tom?
Yeah. Could you please just say again, how do you define the clearance time?
Yeah, the clearance time is just the mean exit time. So then something like that, it's the time that it takes on average for an ensemble of particles to clear the entire length of to transit across this entire piece of tissue.
And so to compute that, you just put a lot of tracer particles in the flow and see how long it.
Yeah, you have the flow profile, then you drop particles, and then just literally calculate how long it takes for them to exit.
And the particles that are stuck in the recirculation zones, they'll never, I mean, they'll never exit.
Yeah, so that exactly, you just pointed out at the mechanism, right? So in this case, if you're stuck, you will never make it out. And that just increases your clearance time. I mean, stochastically, if you add like some noise, then that will help you exit. But then here, because you have the heterogeneity on the tissue orientation or the cilia orientation is providing you that noise, that gives you a mechanism to get unstuck, right? So there's you can connect the streamlines, and that's a physical intuition for why you would want a certain level of disorder. But then of course, if you have too much, then you kind of break the global order and the global connectivity, and then you can see that it's like the domains are completely separate. And then this is what we see. So we have a biphasic relation between the clearance time and the clearance time. So we see that if it's highly organized, it has a minimum at around 0.8, and then it goes up when you have like a higher disorder. But then what was really key here was that since we're able to do this careful measurements of the same parameter from the experimental data, we can see that actually this minimum region corresponds to where the tissue lives, the biological tissue. So then this really suggests that this disorder that we're seeing is something that the tissue builds in to provide robustness for this function. Because by adding this heterogeneity, you're able to clear mucus more efficiently and like have a more globally connected flow. And then, so this really suggests that to design an airway, you just need to worry about like some of these spatial parameters, right? If you're a mouse or a giraffe, it doesn't really matter. The mechanism for clearance is gonna be the same. So as long as you have a level of patchiness and coverage fraction that will allow you to have the streamlined connectivity, then you're fine. And then you're able to generate a globally directed flow. And in this case, if you actually look at the phase diagram of this patchiness, so the separation between the clusters of multiciliated cells, and then coverage fraction, and then you see, okay, here I'm just showing you the clearance time, and then you look at different animals and try to do the measurements for the airway of these parameters, you see that they all cluster around this region. So in this case, because you have some other constraints, you can't have a tissue that is fully covered with cilia, but around this coverage fraction, you see this kink in the phase diagram as well, that if you have low patchiness or the cells in the tissue relatively close to each other, then you're able to generate like kind of the most efficient flow to clear mucus within the constraints you have. So then here I'll stop again and ask if anyone has questions about this section.
I just have a very basic question. So to what, is the heterogeneity affected at all by the fact that you are cutting the, I guess, the trachea and like laying it out?
Yeah, so we were worried a little about that, but since you see that the flow is still globally connected, then that would suggest that the heterogeneity is not affected. And then also people have done experiments where they transplant pieces of tissue, like for example, in a mouse, they take out a piece of the trachea and then implant it in a completely different orientation in another mouse, and they look at what happens with the organization of this, they see that the tissue never really recovers. So that would suggest that any external it's like very robust towards any external perturbations. And then in this case, mostly happens because these are mice that are at a later stage in development. So these are adult mice, but we know this is not the case early in development. So for many systems where you have cilia and multiciliated cells early on, people have shown that if you apply an external flow, you're able to reorient the cilia according to the direction of the external flow. But in this case, we don't think that we are changing anything about the actual measurement or like how it is in vivo.
Thank you.
Okay, so then in this section, I've just showed you like a tiny little bit, right, of what happens if we look at how the coverage fraction and the orientation by multiciliated cells at this tissue scale is designed in a way in a biological system to generate a flow that is tailored for this particular function in the airway. But what I'm doing now in the lab is trying to get a more comprehensive view of how different parameters at different length scales actually feed into this flow architecture. And also, it's been like very like, it's not very well known as well how would actually the physical properties of the fluid would influence these different types of organization of cilia or the activity of the cilia itself, right? Because we were just talking about the airway where we have cilia that are driving mucus, but then in some of the examples I showed you at the beginning, cells are swimming in water. So it's clearly a very different fluid. So this suggests that the adaptations and the organization of the cilia could be very different and then tailored for the particular fluid things are in. And then we of course know that boundary conditions would be important in this context. So in the case of the mouse, it's rather difficult to try and look at what would happen at these different length scales. And also it becomes, in this case, what we would want is to try and make perturbations of these parameters and like see if the predictions of the model are correct, right? We would like to have a way to change, for example, the orientation, the coverage fraction, and see if the predictions of the flow topology are correct. And then you could do this, and the one way to do it would be genetically. So people in the field of cilia have a very detailed knowledge of what molecular players affect different aspects of cilia or different biophysical properties. For example, we know which molecules control the length of a cilium, which molecules could control the density you have at the cell scale, and then also like developmentally, which molecules determine if you have cilia or don't have cilia. So you can imagine like you turn them on and off, and then you can have higher coverage fractions, lower coverage fractions. So we would like to have an experimental system where we can do this. And in mice, it's relatively hard because you need to wait months for them to be able to like generate a mouse that has all these perturbations. And then if you want to look at a different set of perturbations at these different length scales, it becomes impossible. So that's why in my lab, we're using planarian flatworms, where all these perturbations take a matter of days, a week. And then you can have animals that have a disruption in the cilia pattern, and then we can actually experimentally look at the flows you're able to generate and map quantitatively as well from which cilia architecture they're coming. And I just wanna introduce to this audience, because you might not be familiar with planarians, so this is kind of cool and creepy movie. So you can see planarians are famous for regeneration, so that's why people studied them. So you can cut a planarian in like tons of tiny little pieces, and each piece after 2 weeks will generate a whole animal, and you can see each piece is crawling around here. And that crawling around is exactly what I'm interested in. So planarians are able to crawl around because on their ventral surface, they have a carpet of cilia. So they're an active carpet, right? So here you can see an electron microscopy image of all the cilia in the ventral surface of this organism. And then in this live imaging, you can see how they're constantly beating. So then the activity of all the cilia is powering the worm motility. And this is a nice analog for the airway because you have ciliated cells. And then here you can't appreciate really the microscale, but there's also secretory cells. So you have cilia that are moving mucus. So it's very similar to the airway, but it's a much more friendly experimental system to explore. Because you can also, as I mentioned, do molecular perturbations and then ask, okay, how would this affect the ability of the worm to crawl? And then how would this actually, like what is the origin of this defect that we're seeing here? So here I'm just showing you a movie. These are planarians that are actually crawling around. You can see they glide smoothly on the surface. But in these cases where I've depleted certain proteins that are involved in regulation of cilia function, you can see this inchworming behavior. So instead of gliding smoothly, they kind of have to inchworm. And you see a very similar thing in this other case where the worms are inchworming. And in this system, you can also map the flows that the cilia are generating. So here you can see a bunch of tracer particles as well. And you see that even though how they move, at least qualitatively, looks very similar in the terms of just different molecules, the flow has very different architectures in all these cases. So now what we're doing is trying to understand what is happening to the cilia that it gives rise to these different flow architectures. And here you can see a fixed image where you only see the distribution of cilia, and you can see as well that in this case, you can almost have a normal distribution of cilia. So this like defect doesn't make sense, but here you have a very reduced coverage fraction. But then when you look at the dynamics of the cilia that I haven't really talked about, so far, we see that in this case, what is disrupted is probably the asymmetry in that bead waveform. So then even if you have lots of activity, if your bead is symmetric, then you can't propel fluid perhaps, or there's like larger structures as well that are, so here what I'm just showing you is a wave traveling across. So then you also don't see much more of that larger scale metachronal waves. So this is what the type of experiments we're doing in the lab, trying to figure out exactly how these different length scales connect to each other.
And Guillermina, when you look at just these different flow patterns in, say, the control and then this the bottom row, what metrics do you use to distinguish between these? Because actually, I mean, at least to my eye, it's not so obvious.
No, exactly. So that's one challenge that we're trying to work through as well. Like I mentioned in the airway case, it's clear like physiologically the relevant parameters is clearance time. But I would like to like have a more generalized metric, right? To define a flow that has like a higher vorticity versus something that's not. And then like what is the relative length scale of all these different structures? And then so that's something I'm thinking about. And I would love to hear anyone's input on that as well, because I think that's a cool problem that it's not clear yet what the answer is. And then, yeah, for sure, that's something we want to look into. And then I show you what we can do with planarians, but then I'm also looking at, I mentioned also that there's all this temporal patterning in the cilia bead. And this is something that can be readily studied in these free living cells. These are ciliated protists. And then what's interesting in these organisms is that they have as much complexity in functions and behavior as any of these multiciliated tissues that have different cell types. In this case, this is one single cell. But here you can also look at how temporal synchronization between cilia occurs in a cleaner system. So here you have a schematic of paramecium. So paramecium is a single cell that has all these rows of cilia. And you can see these beautiful metachronal waves traveling along the entire cell. So a metachronal wave is just generated by a difference in a phase shift in the part of the stroke of the cilia that neighboring cilia have. So then you can see that because there's this phase shift in neighboring cilia, you overall see a metachronal wave. So I'm really interested in trying to understand what controls the emergence of metachronal waves. Is it through just the hydrodynamic interactions or also through some direct mechanical coupling? Because all these cilia in these cells are coupled by the cytoskeleton. So here you can see an image of these filaments that exist at the cortex of the cell that these cilia are anchored to. So the idea is in these systems, try to understand how through mechanical and hydrodynamic interactions, you can give rise to this cell scale coordinated waves that are traveling around.
And do these planarians have like a nervous system or any,
Oh, so planarians do. Okay. So that's the one difficult part to just understand the physics of metachronal waves. But then it's kind of interesting because then you have another level of control where you can dynamically adjust the direction of your bead or which sections of the tissue are gonna bead versus none. But in this case that I'm showing you here, so in these single free living cells, those guys don't have any nervous system, it's just one cell. So it's just the pure physical constraints. So that's why I think they are a good complement to everything that we're studying in planarians. Because here, yeah, everything you see is just the cilia, or so we think. And then here I'm just showing you another example where you can appreciate the metachronal wave traveling. So I'm interested in, this is just another ciliated protist, again, a single cell, but then here you can really study how arrays of cilia interact with each other and how presumably that coupling can give rise either to metachronal waves in one band or also to the swimming patterns that we observe in the cells. Here we see this is a cell that has two bands of cilia, one here, one here. And then each band has waves propagating across of it. And then these bands are also anchored to each other by the cytoskeleton. So it's another simpler system where you just have two rows of cilia. So then you have a very nice geometry to try and understand what gives rise to these metachronal waves in this case as well. And then I finally want to add a note. So these cells, I'm also interested in them because they are a nice system where you can start to study collective behavior as well. So here the rules of motion for these particles, we know that all the motion, because there's no nervous system, is driven by the cilia. So we know very well what we would expect from the activity of the cilia. And then we can start to understand why would they clump together and what type of macroscopic collective behavior we can see just of these particles interacting. just through their activity. And then finally, I wanna end on this note and say, saying that I'm also interested in understanding these questions of synchronization, right? So cilia are just perfect example of an oscillator. And then here I've just shown you in very different biological contexts where cilia are, but we can also take away all the biological complexity and then just have one single cilium in this in vitro system, and then add activity because we can just add ATP, and then you can see how it starts to beat again. So in this is the perfect experimental system where we can have one oscillator or n oscillators and try to understand how would they synchronize with each other through the different properties of the fluid environment they are in. So with that, I wanna end my talk and then thank both of my postdoc labs where I did the work in the mouse airway. And then if you are curious about knowing more about what we're doing in the lab, here's the lab website, and then I'll be happy to take any questions.
Well, thank you very much for such an interesting talk. So please feel free to ask questions if you have any. Oh, okay, so we have Matt?
Yep. I just had a quick question about that phase diagram where you had all of like the mammals clustered in one area.
Yeah.
And I really just wanted to know what's up with the snake.
Yeah, I honestly don't know what's up with the snake, but that's an interesting question. So we also know that I guess snake physiology is very different as well because it has a very long airway, and then it's like closer to the, like, you know, it's like almost parallel to the ground. And then here I just found like a very low coverage fraction and the clusters being far apart, but I have no idea of why this would be the case. Might be that I just need more data because as well, this is like stuff that I found in certain, you know, in old literature. But I'll be curious about what's up with it because it's also not a mammal, like it's cold-blooded, and then we know that the temperature can affect the beating of the cilium. So there's like a lot of
interesting. There's a lot of confounding variables.
Yeah, there's a lot of room for speculation, but I have no idea really what happens.
Interesting. Cool, thanks.
Hi, thanks for the lovely talk. Really, really interesting. This might be a little bit off topics. That's fine if it is. I'm just curious about what kind of models people try to use to understand these flows and dynamics. So you kind of talked about order parameters at one point, and I was thinking, oh, is it maybe like an Ising or spin glass framework people use, or is it big systems of PDEs for hydrodynamics? Just curious what people do.
Yeah, so the model I showed you is just computational fluid dynamics. So you have in this case the Stokes equations, and then you have different implementations as well of the cilia activity. So in this case, you just introduce a slip velocity at the boundary. So that's the case. But then, yeah, so most of them are just continuum models, I would say, that people use at this. And I'm not so familiar with this literature, but if you're looking at one single cilium as well, you can actually, like, you know, put in like the activity of every individual motor and then how that drives the dynamics. And then, so that's what I would say people look at in this case.
Cool, thanks.
Could you say a little more about this synchronization question? I mean, it seemed kind of like, yeah, you were leading to maybe some ideas, or some results.
I mean, in this case, really, what we know, at least theoretically, is that if you have an array of cilia and then just through hydrodynamic interactions, they can synchronize with each other, or you can get spontaneous generation of metachronal waves. But experimentally, we don't really know if it's pure hydrodynamics or like it's also the mechanical coupling. And we have some good evidence already in this Chlamydomonas algae that only has the 2 cilia. So Kirsty Wan has done a lot of experiments where she had, like, you know, just like one cilia, and then you put it in close proximity with each other, and then you can look how they synchronize and start oscillating in phase or anti-phase depending on the configuration. But then there's more evidence as well that the coupling internally between those two cilia, so they're attached through some filamentous structure, that attachment is important during the swimming because they have to go anti-phase. So then, so that's one good question. I really, there's some literature exploring this computationally, but I don't think there's a lot of experimental evidence of what is important to generate these waves or any other synchronization pattern. And then also another thing that it's unclear is like, why would you need metachronal waves? Like we see them everywhere in biological systems, but we actually don't know how, like by tuning, for example, the wavelength, would that affect like, you know, the flow structure or like its properties. So then that's another thing that we can have access to experimentally in the systems. Then hopefully there will be more and less speculation at some point soon.
Do we have any more questions for our speaker? All right, I guess we will just wrap up here for today. I'd like to thank our speaker, Gilmina, again, and everyone for participating. Hopefully see you all next time.
Bye.
Bye.
Thanks, bye everyone.
Thanks.